I'm going to talk a little bit about the sort of my life prior to CanDIG. And Open Tree of Life is the project that I was a PI on for the years that I was at Duke when I was in the States, and I still do some contract development for them. Yeah, with that, I'm going to tell you about Open Tree. So the aim of the project was to put online a sort of best current scientific estimate of what the tree of life looked like. So a tree that was complete, that was online, and that also was being constantly updated. So part of why we needed this was that despite an enormous amount of funding being put into phylogenetics and tens of thousands of papers being published that contained phylogenetic trees, no one had done a synthesis of that knowledge and put the tree somewhere that all of the fields of science and education and museums that wanted to use the tree of life didn't have a resource where they could go and just say, like, tell me about the tree of life, or let me pull down part of it. So that was the motivation of the project. So the way we did that was not to go to GenBank and get all the genetic data we could and build the giant tree, because there were lots of groups already doing that for various parts of the tree of life. So what we wanted to do was take all of those publications where someone with expertise in a particular group had published, the best estimate of what the tree was, and combine all of those. So combine published phylogenies with taxonomic data, so taxonomic hierarchies that are available on the web, and put all that together into a single estimate of the tree of life. And once we had that, then we could let people ask questions about it and use the data in ways. So now putting in sort of analysis services, so like conflict, how does my tree compare to yours or to some other tree, to be able to say, hey, I want to do some research project with these species and I need the tree that relates them. Or somebody coming and saying like, hey, the tree is really terrible for this group that I am really interested in, but I have a better one. Like I have an estimate and can I upload that and make yours better? So these are the sorts of questions that we were trying to enable by putting this tree online. And with that, the most of the rest of this talk is actually going to be live demo. So cross our fingers. So I'm going to start at this is the mammal node. Mammals, taxonomic group we should all be fairly familiar with. So we have a very minimalistic and not too pretty tree viewer. And I'm going to say more about that in a bit. But one of the things that we really try with the project is to do a really good job of provenance. So where does our data come from? Why are we saying these things about the tree? So for every node in the tree, we have links to all the taxonomy inputs. So NCBI, the Global Biodiversity Project, other taxonomies that say something about what they mean by mammalia. You can download the tree. And then we have a list of the supporting or input phylogenies that support the edge leading to mammals. So if we browse to an alternate node, so this is a case where this node doesn't have a taxonomic name. If you go to a taxonomic hierarchy, there's no official name for this node. So we give it this X plus Y label. So in this case, there's no links to taxonomies because this only exists in phylogenetic trees. But we see here that we have trees that both support the existence of this node and trees that conflict with the existence of this node. So that's what I kind of mean by saying, putting a lot of information about why we're showing what we're showing. So for any of these, I can click on one of these input trees and I go to the curation interface that we have for phylogenies. So we group these by publication. So everything, sort of the basic unit in that database is a scientific publication, which may contain one or more phylogenies. So there's publication metadata, there's a list of the trees that are there. There's a status about whether or not the tree is currently included in the synthetic tree or not, or if it's not, this one would need additional curation before we could include it. A huge amount of the curation time is spent on mapping the tip labels on the trees to taxonomic names in databases. And so there's an automated process for curators to do this mapping. Various analysis tools. So if we pick one of the trees in that study and we say, how does it compare to the synthetic tree of life? I can get this little report. I can open the tree. And that tree is now labeled as all the branches that agree with our synthetic tree or disagree with our synthetic tree. And so we're looking at putting in additional analysis tools for any given study. And then there's a history tab. So we can see for this particular study, it's had 3 different people who have made edits to it over periods of time. There's a comment about what everybody changed. And then for any of those edits, we have a diff view. So when I click on that, where I go is GitHub. So we use GitHub for authentication for all of our curation, and all of the study information or tree information is stored in JSON files in a repository that's called the file system. And so this allows us to make use of existing authentication and not have to roll that ourselves. And it also gives us full history of every edit that anybody has made to any of our trees so that people can see. You can also, if you want to use the underlying database, this is the canonical database. You can just clone this repo, do whatever you want with it. So we want to make it as easy as possible for people to use and reuse this data. Having this in GitHub also gives us a nice way to build curator profiles. So it's really easy for us to tell, this is me, how many studies have I contributed, how many have I edited. All of our feedback goes through GitHub issues, so we can track all of that information relatively easily without having to have a big user database of our own. So if I go back to, let me go back to our mammal node. So I had sort of mentioned that this user interface is not that pretty, and that wasn't really an area of the project that we really focused on very much. We focused on assembling the data and providing really good APIs for people to access it. So one of the awesome things that people have done, another group has done with our data, is create a really nice fractal viewer of the tree called OneZoom. So this allows us to browse the phylogeny at really, really gory detail. So the structure here is the open tree of life, and then they're annotating it with images and other metadata from Wikipedia, Encyclopedia of Life, and other resources. So in terms of, how well is the project working? The community curation part has not been as successful as we had hoped. And there are reasons for that, that I could talk to if anyone was curious about that. We've got about 5,000, a little under 5,000 studies in the database, about 50 people who have curated, and about 11,000 commits over the length of the project. But I think not so much, so the data deposition piece has not been as successful as we hoped, but the sort of use of the tree has been like really, that part we feel a lot happier about. So I already showed you the OneZoom piece. The tree's been used in a number of museum exhibits. And then this one here is sort of in terms of scientific use. A group went and built an R package that wraps our APIs. It's part of the R OpenSci infrastructure. So here's a list of all the scientific publications that have used trees from OpenTree that they got through the ROTL package to do scientific analyses. And this is the kind of thing that we really wanted to enable with OpenTree that really wasn't possible before because there was no single resource that had phylogenetic data. So this obviously was a large project. The first round of funding was 13 principal investigators across 12 institutions. The current funding is 2 PIs, and I have moved from being PI to a developer after leaving the US. But there's just been a huge number of people, students, devs, who've worked on it over the years, and all with funding from the National Science Foundation. And yeah, that's all I have, and I am happy to take questions.
Fantastic, thanks. So who has some questions for Karen? I have a first question. So it seems like it was the project is a lot of, there's tech work and a lot of governance work on top of the curation work. Like somebody, I guess, has to decide what goes into the synthetic tree. How does that work?
So initially, when in the first round of funding, when we had like 12 different universities involved and an enormous group of postdocs and grad students, the postdocs and grad students played a really huge role at trying to track down what were the sort of best phylogenies in their area, whether that was birds or fungi or plants, and try and get that data in. So that was very manual initially. Now we actually allow curators to add trees to the synthetic pipeline. And then when we build the tree, we have a lot of sort of checks and tests for, oh, we added this tree and it created a lot of havoc. So maybe we need to take a look at that. Maybe there's some curation error, or if, you know, for example, because taxonomic names are not unique, you can have a case where somebody maps like a frog to a plant name, which then causes all kinds of chaos down the line. So there's just building the synthesis, there's just a lot of checks about like, is the result that we're seeing at the other end sane? But we've been moving more and more to letting curators, you know, make those like, yes, this, I want to, if this tree passes all the quality checks, then yes, you can just tell us to put it in the synthesis.
Very cool. Who else has some questions for Karen?
I was wondering, did we ever figure out how to ingest trees more quickly, or do people still have to like write Nuix strings and import them?
So the import is still primarily Nuix strings, which is sort of the canonical way that this community, it's a parenthetical notation for trees because this standard appeared before JSON was really a thing.
And there's also some good reasons why JSON or tree like Nexon and NexML never really took off. In terms of
there's a long history there. Yeah. The one thing we are working on right now is what we're calling script managed trees, because the curation interface fails once you get to a certain size of phylogeny. And so we've got a prototype where you can push large trees directly through the API rather than have to go through the graphical interface. And you can do sort of the OTU mapping and things like that, sort of on more dev type tools rather than the UI. So to get some of the sort of huge mammal and bacterial and fungal trees that have come out the last little while.
Yeah, like I guess like, yeah, you'd have to have the and I guess now that everybody is supposed to be putting things into like putting the data should be available with the paper.
Haha.
Yeah. These are the reasons why that phylogeny part of the community thing has never gone as well as expected.
Yeah, this community is weird in that the sort of norm for data sharing is like, of course, so the basic pipeline is you get genetic data and you align those sequences, and then you use some sort of software package to build a tree, right? And the norm for data sharing is, here's the gene sequences that I used, and I put them in GenBank, and maybe here's the alignment. But as far as the actual output, like the actual tree, the most common way for people to publish that is as a picture in their paper. And so that's been a challenge, is actually getting those tree files, which are the only thing that we can really do anything with. So it's still this.
Yeah, the problem is like they're like my connection to these projects has always been like which was frustrating, because what was the first hackathon I met you at wasn't like a decade ago now. So these
I think it was more, Daisy.
Yeah, these problems are still, as far as I can tell from what you're saying, extant problems now. One, that there is the reason that we do it in tree file, like pictures, is because people like to decorate their trees for publication with a bunch of notations that is basically impossible to put into a tree file in any kind of real way, because we don't have we don't have a really standardized decorated tree format, for like enhanced metadata of trees. So I've half-assedly tried to manage projects for that at various hackathons and never gotten anywhere, because I am just not that dedicated to these problems. And then two, I wrote like at a previous Open Tree of Life hackathon, I like worked on a project to like make tree strings, like Nuix strings out of pictures, and got like maybe 2/3 of the way there. And then you know how those things are. It's like you're never done, and 2/3 is not enough to really do anything with. It's just frustrating, like how much we're never going to finish this at this point, like these holes, still same holes. Anyway.
Does anyone else
this is just me chatting with Karen now about genetics.
Does anyone else have any questions for Karen? All right. Well, let's thank Karen. And of course, you can always talk to Karen on Slack. And introduce our next speaker, Courtney Goslin.